Welcome to The Explainer. Today we are diving into the absolutely wild world of genetic engineering, but we're going to look at it through a really cool lens, molecular carpentry. Think about it. Just like a carpenter building a house needs very specific tools to cut, shape, and glue wood together, scientists have developed a microscopic toolkit to cut, modify, and glue DNA itself. It is precision engineering just on a cellular level.
Here's our roadmap for today. We'll start with a brief history, walk through the 5 key steps, open up our enzyme toolbox, check out vectors, our delivery vehicles, and then finish up with the real-world impact. Let's get right into it.
Part 1. A brief history, the DNA blueprint.
So at its core, genetic engineering is really just a laboratory process used to alter an organism's DNA makeup. Whether you're changing a single base pair, deleting a huge chunk of code, or pasting in a brand new segment from a totally different species to get a desired trait, it is literally the ultimate microscopic carpentry project.
And you know, the speed of discovery here is just mind-blowing. In 1951, genetic engineering was literally just a made-up term in a sci-fi novel. By 1953, Watson and Crick gave us the double helix. But then, the 1970s hit, and boom, an absolute explosion of progress. In 1972, Paul Berg creates the very first recombinant DNA molecule by combining monkey virus and lambda virus DNA. The literal next year, Herbert Boyer and Stanley Cohen make the first transgenic organism using E. coli. And just 1 year after that, Rudolf Janisch engineers the first transgenic mice. We went from sci-fi to a complete biological revolution in just a couple of decades.
All right, part 2, the 5 steps, engineering genes.
So how does this actually work? Well, there are 5 universal steps to this whole blueprint. First, you've got to identify your target gene. You have to know exactly what trait you're trying to express. Second, you isolate that specific gene. Step 3 is cloning and production, where you basically make identical copies of it. Fourth, you introduce that cloned DNA directly into your host genome. And finally, step 5 is expression, where the host actually starts producing that new protein or trait. Plant, animal, bacteria, it doesn't matter. It's the exact same sequence every time.
Moving on to part 3, the enzyme toolbox, our molecular saws and glue.
To actually execute this microscopic blueprint, we need our carpentry tools. We've got 4 essential ones here. Restriction enzymes are our molecular saws. DNA ligases, that's our glue. Polynucleotide kinase, or PNK, is what we use to modify the ends of our DNA strands. And alkaline phosphatase acts kind of like safety caps for our biological delivery vehicles. Let's talk about those saws first. Now, not all saws are created equal. If you look at the cleavage patterns for type 1 and type 3 restriction enzymes, they cut completely randomly, sometimes 1,000 base pairs away from where they actually bind. You definitely cannot do precision carpentry with a saw that jumps around like that. That's why type 2 restriction enzymes are the absolute go-to choice. They are incredibly precise and cut exactly at their specific recognition site. So how do these type 2 enzymes know exactly where to make the cut? Well, they hunt for DNA palindromes. These are specific sequences with twofold rotational symmetry. Basically, if you read the 5' to 3' strand and then read the complementary strand in the exact same direction, it's the exact same sequence. For example, the enzyme EcoRI is constantly scanning for the specific sequence GAATC. When it finds it, it makes a perfect, precise cut.
When these little molecular saws do their job, they leave behind one of 2 types of ends. You have blunt ends, which is exactly what it sounds like, a clean, straight, equal cut right across both strands. But honestly, what scientists really love to use are sticky ends. This is an uneven cut that leaves a tiny overhang of unpaired nucleotides. It's essentially molecular Velcro. These unpaired bases naturally want to stick to their matches, which makes dropping in our new, foreign DNA vastly more efficient.
Okay, so once those sticky ends find each other, you have to permanently seal the joint. Enter DNA ligase, our molecular glue. Discovered independently by 5 totally different labs back in 1967, this enzyme uses energy, like ATP or NAD+, to form a rock-solid phosphodiester bond between the DNA fragments. It perfectly seals up any breaks and permanently locks our new genetic sequence right into its new home.
But there's a catch. A super common problem in molecular carpentry is that your delivery vehicle might accidentally just glue itself right back together before you even load the new gene inside. So we use alkaline phosphatase. This clever prep tool pops the 5' phosphate groups right off the cut DNA ends. And without those phosphate groups, the ligase glue literally can't seal the empty vehicle shut. It basically guarantees our target gene actually makes it inside.
Which brings us to part 4. Vectors, the delivery vehicles.
Once your DNA is cut and glued, how do you actually ship it? Our most common delivery truck is called a plasmid. In nature, a plasmid is just a tiny, circular ring of extra DNA hanging out in bacteria, usually carrying handy survival traits like antibiotic resistance. But scientists have totally hot-rodded these natural rings. By adding things like multiple cloning sites, which act like loading docks for our restriction enzymes, and specific selectable markers, these plasmids become the absolute perfect customizable trucks for hauling foreign DNA straight into a new host cell.
Now, not all delivery trucks do the same job. They generally split into two categories. You've got cloning vectors, which are basically microscopic photocopiers. Their only job is to churn out massive amounts of identical copies of a target gene. On the other hand, you have expression vectors. These are full-blown biological assembly lines. They come fully loaded with extra genetic instructions like promoters and start/stop codons, telling the host cell, hey, read this DNA and actually manufacture the protein.
Plasmids are incredibly useful. But what if your gene is just way too big to fit? That's when you call in the heavy-duty rigs. Bacteriophages, which are essentially viruses that target bacteria, can carry much larger inserts by swapping out their own non-essential viral genes. And if you need to move truly massive payloads, like giant chunks of a eukaryotic genome, you use artificial chromosomes. Things like yeast artificial chromosomes, or YACs, can haul a staggering payload of up to 1 million base pairs at a time. It's incredible.
And finally, Part 5. Real-world impact, molecular carpentry in action. So why does all this microscopic cutting and gluing actually matter? Well, this science has completely reshaped our modern world. Take 1982, for example. That is when the FDA approved human insulin for treating diabetes. That was the very first consumer genetically modified organism, built entirely with these exact recombinant DNA techniques, and it absolutely revolutionized medicine. Then in 1994, the first GMO tomato hit grocery store shelves. By 2005, engineered crops like alfalfa and sugar beets were everywhere, totally transforming global agriculture to be more resistant and productive. Which brings us right to the edge of tomorrow. Think about it. We've gone from just figuring out what DNA looks like to literally cutting, pasting, and mass-producing genes that save lives and secure our global food supply. And as biotechnology pushes past basic recombinant DNA into this wild new era of incredibly advanced genome editing, you really have to wonder, what exactly is humanity going to build next with our molecular carpentry tools? The blueprint is literally in our hands. Thanks so much for joining me on this explainer, and I hope this sparks your curiosity to keep exploring the amazing world of genetic engineering.